Good afternoon to everyone. I'd like to thank the organisers for giving ELIXIR the opportunity to showcase some of our groundbreaking projects that the ELIXIR members are working on within their nodes. For those of you that are not acquainted with ELIXIR, its aim is to bring together life science resources, database tools, compute, interop services and teachings, alongside use cases from communities under a single distributed infrastructure. Our speakers today will try and give vignettes from the infrastructure landscape, moving from 3D structure beacons, proteomic analysis, ontology for COVID-19, and a tools platform that can provide COVID-19 solutions, some update around federated human data sharing, and then ending with making data fair by design. So we have 6 talks and a very tight schedule. So I'm going to start with Samir Velankar from PDB, who's based at Emble EBI in Cambridge, and he will present on 3D beacons, an integrative distributed platform for fair access to experimental and predicted micromolecule structures. Thank you.
Thank you, Jen. So I'm going to present, as Jen said, 3D beacons, which is a new project we started this year, and I'll tell you where we are with the project. But the idea is to make experimental and predicted structures available through a common platform. So let's start with experimental structures. And that spans quite a scale, right from atomic resolution structures to whole cell and whole organ studies. And there are various techniques used for that as shown here. And there are multiple data resources and archives for these different types of data. But what we want to achieve finally by getting all these structures is to be able to use this molecular level data and its biological context and additional information to convert that data into knowledge. We want to understand the biology behind a particular protein or a particular complex. And that's the goal we have for archiving all this structured data. So how can we achieve that? As part of the Worldwide Protein Data Bank, we at PDBE are involved in archiving the macromolecular structures and managing the protein data bank. But there are many different specialist databases which take this macromolecular structure information and analyze it and add annotations to a particular residue or a set of residues and to enhance the information that's available from these structures. So the PDBE Knowledge Base Consortium was established to take all these different specialist data resources, bring them together and collect a set of annotations that we can then interconnect and create a knowledge graph. And this is what PDBE KB offers. There are various types of annotations that are integrated in PDBE knowledge base. So how does this benefit for example, PDBE KB itself? We can provide better service to our users through this enriched data. The partner resources also benefit because they get better visibility. There is increased impact of their work and the annotations that they provide, and they reach many more users. And of course, the users benefit because they can access this diverse data from these niche data resources through a unified platform and also through standardized data formats. And so you don't have to learn different types of APIs or how to access data. And at the moment, there are 26 resources collaborating to provide this information from 9 different countries. So can we answer scientific questions if we do all this and bring all this data together? And these are just some example questions that we are able to answer. And it goes from something as simple as downloading structures for a given protein to analyzing conformations of proteins or trying to find binding sites for ligands. So this is COVID-19 protease, and you can see the fragment screening experiment has provided a rich information on where ligands bind. So there are multiple scientific questions that we can answer through this knowledge graph. But is that sufficient? No, it's not, because in the experimental archives, we have very small amount of sequences represented. And so in terms of sequence coverage, PDB structures are minuscule. Although it does provide a lot of PFAM families, and which means we can transfer some of these annotations to other sequences as well. But we cannot ignore the fact that structure prediction methods have improved quite a lot. And actually they now are used during structure determination. And the models can provide improved coverage of sequence space. But at the moment, that data is available in different data resources. So here are some data resources that provide these predicted models. And each of them provide their own API. And so as a user, you have to then know where to find the appropriate model. So this is the motivation behind starting this 3D Beacons project. So our goals are to design and implement an infrastructure that brings together all these different data resources on a common platform. So we want to register resources that provide structured data. We want to develop a standard with them for data exchange, create a common data access mechanism, and as a result, we hope that there is reduced fragmentation and users will benefit from getting access to all the structured data that is available in all these different resources. So what are the example use cases where we can apply this? For example, if you are interested in effect of variants and want to infer how the variant might affect structure or function, having a model of that particular sequence is very useful. Or if you want to do tractability assessment, it's very useful to have structure models for the human proteome. Or as I said already, you could use some of these structures in determining new structures using cryo-EM or X-ray methods using molecular replacement. So we started this project in this year, early in January, and the idea is to establish hub and a registry. So the registry will contain all the different data resources. The hub will allow programmatic access and a user interface to all this data and present all this information to users. The individual data resources will be beacons. They will host a API client, and then the hub can query that and provide information to users. So these are all the different teams that we hope will join, and also different data resources and applications that can make use of this structured data and the 3D Beacon platform. So where are we now? We are working on a prototype, and that's being co-developed by the PDB-ECCB team, the Swiss model team, Genome 3D, and Foldex, and Uniprot is also contributing use cases and will provide feedback. We are also developing software to allow out-of-box implementation of a beacon, and which means other data resources who are interested can establish a beacon very quickly. And currently we are following these use cases. So Uniprot accession and a residue range, and is there structured data available for that? Next, we will move on to having an amino acid sequence and then provide structures. So you can access all the code here, and this is the development version available on that website. And I want to invite people to participate in the biohackathon. We will be there. And the idea is we will have a working end-to-end prototype by then. And we want to work with users as well as data providers to enhance this whole platform and 3D beacon infrastructure. With that, I will like to thank all the collaborators we have who have worked very closely to establish this infrastructure and the PDB knowledge base. And I'll take any questions. Thank you.
Thank you, Samir. That was a great presentation. Please post your questions in the Q&A. I don't see any at the moment, but I'll have a question, Samir. Is there a limit to how many resources can be part of this beacon?
No, not at the moment, but we are also making sure that any resource that joins also has quality information with the structures. So we can provide both the coordinates as well as the quality of model to users. And so in the software package I discussed, where you can establish 3D beacon very easily, one of the components is the quality criteria and quality validation of the predicted models.
So I have another question here from Hiresh Dong. Are you also working on the structural domain, like in the structure of how many and which domains are available?
Yes, so the use case I described was a uniprot accession and a range of residues. And so for a given domain, you can imagine providing a uniprot accession and a range of residues and then querying all these different beacons to see if there are models available. And some of the data resources will provide structure models for individual domains rather than the whole protein.
Okay, thanks, Samir. We'll move on to the next speaker because of time, but please keep posting your questions to Samir. You can answer them online. The next speaker up is Jose Ramon Macias from the Biocomputing Unit from the Spanish node. He's going to talk about 3D BioNotes COVID-19 edition, bringing together structural and functional information on SARS-CoV-2 proteome. Please take it away, Jose.
Yeah. Hello, everyone. Let me share my screen. I think you can see my presentation now.
Yes, yes, continue.
All right. Hello, everyone, and thanks the organizers for letting me present you 3D BioNotes, especially our new edition that we have made a big effort to compile structural information and functional information about the SARS-CoV-2 proteome. 3D BioNotes is a web platform that integrates a set of web services and an interactive 3D viewer to provide a unified environment in which biological annotations can be interactively studied in their structural whole context. Some of the tools that are available, for example, through a simple API, that others, like the ones that we provide to calculate protein-protein interaction networks, can be accessed simply by filling in a form in a separate section of the website. Even the user can provide their PDB or ENDB entry identifier that is interested in, or even can upload their own structural models that they are studying locally in their lab, and also provide locally available annotations if they like to analyze in the same integrated way. But also, many of our users can directly from links that are kindly provided by the electron microscopy database. In every entry, the user has these kind of links that open directly our 3D viewer. As I mentioned, we have made a big effort for compiling many structural information about COVID-19. There's a big effort also all around the world to provide the structural data for these viral proteins, many including cryo-EM studies, which is actually the main background of our group, and also other experimental techniques like X-ray, NMR. Some of them has been also in the previous presentation, together with modeling and computational predictions. So what we have done is dedicated a new section of our website with the intention to collect and present in a single place all structural information relevant to the SARS-CoV-2 virus. In this new section, apart from some interesting examples that we have in the top of the section, we have added simplified schemas of the proteome of the virus, and more recently, a nice infographics that can be used as a kind of index with links to the corresponding subsections of every protein in the virus. Then we have a panel with a brief description of the protein, its role, for example, the role in the virus life cycle, some about the biochemistry and so on. And these panels have contain all known structures organized in 5 sections. In the PDB section, we have all the atomic models published yet, including, for some of them, when available, validation data provided, for example, from PDB Redo or Isolde. These images act also as links to show every entry in our 3D viewer, I will show later, but also provide additional details and external links and so on. In the MDB section, volume maps from the electron microscopy database are collected in a similar way. And such as some structures contain more than a singular protein, they are grouped together under an interaction section. And these interactions can be about protein-protein interactions with other proteins in the virus or with proteins from the host. And also can be interactions with ligands, with small molecules that have been identified in drug screening studies. For some minor viral proteins, there's not experimental structural data yet, so we have collected some computational models and predictions that may help to have an idea of the possible structures of these unknown or unstudied proteins. And for completeness, related proteins from the coronavirus, like SARS-1, MERS, and so on, are also collected and displayed in this related structures section. The 3D viewer is probably the main component of the 3D Bionodes platform. And it's composed of 2 panels. In the left-hand side, we have the full interactive 3D display. And in the right-hand side, we show all the relevant annotations for that structure. There are different types of annotations organized in separate tracks with boxes, different symbols representing each annotation along the axis of the sequence. And as you may see, and by clicking any of these boxes representing an annotation, all the residues associated with that annotation are highlighted, also in the protein structure or in the alignment sequence, which is bottom left, in the atomic structure. At the same time, these residues will also be highlighted with vertical yellow bars, so it is easier to locate these positions in relation with other annotation types. Additionally, a panel will pop up with more detailed information about the annotation, including links to to the origin of the data source. In this effort for the COVID-19 edition, we are able to provide a set of annotations. For example, these functional mapping tracks allow to locate the residues that likely constitute ligand binding sites and protein-protein interaction binding sites. This covers homo and hetero oligomeric interactions among SARS-CoV-2 proteins and other SARS-CoV viral or human host proteins. And yeah, this comes, well, in the pop-up panel, you can go directly to the source of the data, you see. We also provide functional mapping of ligand binding sites from the same origin, and some annotations that are related to drug screening experiments that have been observed, studied in the XM fragment screen of diamond synchrotron in Oxford. Yeah, where the track is expanded, one row per different ligand will be displayed, showing the location of interaction residues, which the ones corresponding in the actual structure in red. So you can easily locate those residues in the structure. Also, we have a special track for variants, genomic variants that have been collected by the China National Center of Bioinformation. Here you can see different rows of aggregated data with several mutations at certain position. And yeah, most of them are uncharacterized, but yeah, you can refer directly to the source of the data also. We provide also cryo-EM map validation scores or data for those cases where there is an electron microscopic map. We show local resolution scores that are calculated in our own group, applying deep-res and mono-res algorithms. If there is an atomic model available from PDB that is fit in that volume map, we also can calculate MapQ scores. The local resolution values are displayed per residue using the color code, which usually meaning which cold colors indicates higher and hot colors lower local resolution. This way, it is quite easy to find interesting regions of the structure with similar resolution values and get a better understanding of the flexibility and conformational dynamics of the structure. So, yeah. And yeah, finally, let me show the team that is of the biocomputing unit of the CSIC for the cooperative work in this special situation during the lockdown, and thank them all. So thank you all for the attention, and glad to get some questions from you.
Thank you, Jose. That was a great talk. I just have while people are posting questions, hopefully they do, I have a question. The diamond synchrotron, which proteins are they focusing on from COVID-19? Do you know the drug discovery?
Yeah, we have right now we have data for just one experiment, which is the main protease. In this case, was the target of the experiments. And we are actually working now in also updating our data set with another experiment that they have just carried out about NSP3, which is the ligand, I think, ligase, I think.
Okay. I don't see any more questions at the moment, but I'm sure people will post them.
Yeah, I will.
Because of time, thank you. Because of time, we'll move on. I'd like now to introduce Deborah Caucheteur, who's from the Swiss Institute of Bioinformatics, from our Swiss node, and she will be talking about COVOC, a COVID-19 ontology to support literature triage. Take it away, Deborah.
Thanks, Jen, for this introduction. Hello, everyone. Here I will talk to you of COVOC, a project developed in collaboration with Ellen Parkinson's team at the EBI. Sorry. The COVOC project started in March 2020 in response to the pandemic crisis, and our objectives were the creation of a new ontology fully dedicated to COVID-19, and in the second time, the conception of a curation support literature tool for researchers interested in COVID-related information. We started with the creation of a large Google spreadsheet, split it in 9 axes, where we added many terms related with COVID-19, and then we decided to share this file with the scientific community to expand our vocabulary. At this time, Ellen Parkinson's team at the EBI proposed us to collaborate for the conception of a new ontology, which named COVOC. COVOC was developed as an application ontology from template files in order to allow collaborative development of the ontology. The template files were created from the original spreadsheet of concept through manual curation. And the aim was to include as many terms as possible from existing ontology resources, combined with more specific novel terms created for COVOC. Terms that are both novel to COVOC and imported from other ontologies have additional annotation properties to aid users, including a preferred label, cross-references to both OBO ontologies and other vocabularies, internal identifier, and synonym. The templates were then converted directly from Google Sheets to an ontology file using the ontology development kit and robots. And finally, Protege, an ontology editor, was used to check COVOC after running the pipeline. As results, COVOC contains 543 concepts, plus more than 1,000 synonyms, thanks to some import from 20 existing ontologies, spreading over 9 axes, like drugs, organism, or disease and syndrome. This ontology was very useful in the development of our curation support literature tool, and I will explain you why. The process behind our curation support tool starts with a terminological entities treatment. We split COVOC ontology by axes, the same defined in the initial Google spreadsheet. We separate concepts with their related ID and synonyms in 9 distinct files. Then we screen collection, Medline, free full text from Europe PMC and Code19 from the LNA lab with all those terminological entities. We create annotation for each document when a term is matched, and we put it in a database. Here, as you can see right here, in this paper, you have an example of annotations, one object for one annotation. In that case, the concept form is source, it's the matched term. And finally, this database is imported in a search engine to return ranked document as response to a query sent by a user through COVID-triage, our public tool. Before showing you the interface of the tool and talking to you of an additional information related on it, let's have a look on some statistics about the terminological entities recognition. You can see that we have an average of 8 COVOC annotation for the Medline collection, 140 for the free full text of Europe PMC, and 319 for coordinated collection. These statistics evolve every day because our collections are updated daily, followed by our annotation process. A last information is required to understand how COVID triage works. At the same time as we were working on the COVOC project, we took part in the competition Track COVID run by NIST. During 5 months and through 5 runs, participants worked with a collection of papers related to COVID-19, and in parallel, a list of topics was provided. These topics represent queries. The participants have to submit for each topic a list of 1,000 papers among the collection, which are ranked by relevance. And at the end of each round, some metrics are calculated based on the relevance judgments file. We have tested several strategies, and one of these relies in the document re-ranking based in the density of terms in COVOC axes. For each query, each topic, we assigned manually one or more COVOC axes. In this example, at the query, what is the origin of COVID-19, we assigned organism and geographic location because the term origin could refer to host or vector organism, but also to countries or city where COVID-19 was discovered. Paper will be re-ranked to push at the top those with annotation from organism or geographic location axis. We have improved several metrics, and that's the reason why we use these re-ranking strategies in COVID triage. This is the COVID triage interface. You can write your query, choose an axis in reference to the re-ranking strategy presented just before, select the collection you want to screen, and so on. Just an example here to show you the results interface. The query was spike protein, a protein related to the COVID-19 mechanism, any preference about the axis, and we are looking for information in the Medline collection. The articles are ranked with a score relevance. You can see the number of COVOC annotation found in the abstract. Your query terms are underlined in the title, in the text, and COVOC entities are in purple. Some improvements are in progress, like the integration of a viewer for the free full text collection. With this viewer, the users will have access to the paper directly into the interface. COVOC annotations will always be highlighted in the text, and it will be possible to choose what axis you want to highlight. At the end, users can decide to download one or many publications that they consider relevant. In the future, we have to think about some points. First, the maintenance. The ontology was developed from spreadsheets to make it easier to maintain without the need to be an expert in using ontology tools. We have to find a quick and efficient way for the next updates. Also, it's really important when you create an ontology to promote the interoperability. That's the reason why COVOC will be submitted soon to Ober Foundry. Another point we should explore is the consistency of axes. We should try to align our distinct axes with the curator's needs. Axes are not all homomorphic. For example, in the organism axis, there are vectors like pangolin and virus like coronavirus. Both are organisms, but from a curation point of view, it's totally different. Then, about the next collaboration, we are in discussion with the OLS team, and soon, thanks to Europe PMC and Platzi, we may be able to offer to COVID triage users some new contents like preprints collection. This project is based on a strong collaborative work. Despite the pandemic crisis, we have validated several steps since March. Today, we have an official release of COVOC ontology and the first version of COVID triage. We are still working on several improvements and with new partners. we would be happy to receive your comments and idea to improve it. I would thank my director, Patrick Ruch, and colleagues who work on this project, as well as our collaborators at Platzi and EBI for their involvement, and the audience for its attention. Thank you.
Thank you, Deborah. Thanks for a great talk. We have a few questions here. To start off with, Ciro wants to ask, in your experience, what was the biggest challenge when cleaning up the list of terms coming from many sources when building the template spreadsheet?
In fact, the Google spreadsheet we created by ourselves at the first time, and then we distribute it. And the manual correction was made by the EBI. So maybe Paola and Zoe, who are in the audience, could give a better answer than me via the discussion forum. But maybe I think the biggest challenge in the creation of the spreadsheet was to make the cross-reference.
Mm-hmm. Okay, great. John Hancock wanted to ask, does your CV contain terms that are not in OLS ontologies? And if so, will you be feeding them back and incorporating them into that?
I think Ellen Parkinson's gave an answer.
Yeah,
because it's on the ontology part, so it's more on the side of EBI.
And then there's a final question. Although Foundry focus on domain-specific ontologies, I've not heard much on them except in application ontologies. Are you planning to, oh, this is a very similar one, submit them to infectious disease ontology?
Oh, in what aspect of COVID-19?
So I presume that's from Helen, yes.
Yeah.
They will be fed back. So I have a very quick question around the competition that you took part in.
Yeah.
What do you think was the benefit of taking part in this competition, and what did you learn from it?
In fact, I think the track objectives, there is two main objectives about track. The first was to evaluate search algorithm and systems for helping scientists to manage a growing corpus of scientific literature related to COVID-19. And the second main advantage to take part of in this competition is to discover different methods that will assist with managing scientific information. At the end of each round, you have access to a report of each team, and you can see what the other do, in fact. And I think it's a good way to discover new methods. Okay,
perfect. Because of time, we'll move on. Thank you very much, Deborah, for answering those questions.
Thank you.
Thank you. We're going to move on to Bjoern Gruening from the University of Freiburg, who's from the German node and is part of the tools platform EXCO. He's going to present on the tools platform and highlight where solutions for COVID-19 research could be benefited from the platform. Are you there, Bjoern?
Can you hear me, Jen?
Ready? Perfectly.
Can you also see the slides?
I can, yes,
Perfect.
They're fine.
Okay, yeah, I would like to introduce you to the ELIXIR tools platform. And we would like to introduce that with a practical example on COVID-19 research, which we spent most of the years of this year on different aspects in the tools platform. So this is more or less a timeline. So we started actually very early on, so in February already, to get ready with compute resources, because we have seen that many more requests were actually going in, so both computational wise but also training wise, so that people wanted to migrate from classroom teaching and training to online training. But we also saw a need in data analysis, so we were ramping up all our different aspects of the tool platform, which contains bioconda and biocontainers, but also tool integration into already existing platforms like Galaxy. We have worked on annotating tools so people can find more easily COVID-19 related tools and resources. We participated in a few hackathons actually, but one of the biggest one was a biohackathon that was also co-organized by ELIXIR. At some point, we started to actually develop e-learning material specifically for COVID-19 research, drug design and drug discovery, but also analyzing the genome. And the Workflow Hub team, so that's a project that we wanted to start actually later this year, but due to the urgent need, Carol and Frederick's team actually started then also very early on to create this new resource, Workflow Hub, where we share workflows. And nowadays we are at the stage where we still develop workflows, but these workflows are now automatically tested and benchmarked so that we can address the urgent need and the request by our users. And I would like to guide you through all these projects a little bit, what we have done, what these projects are in the next minutes to explain you more or less what the tools platform in ELIXIR is doing. So let's start with the basic work of Conda and biocontainers. So no matter what you do, if you want to run computational analysis, you need to have tools. So some binary sets needs to be installed. And the ELIXIR tools platform, or one part of the ELIXIR tools platform is the container or the biocontainer community. So we maintain, we create, and we develop more or less 8,000 containers to all different kinds of aspects of biomedical research. And these containers are all registered in this registry here. So there's a web form where you can search your tools, and you will see ultimately such a list where you can see your different resources. So there's a tool available in Conda, available in Docker, even Singularity, and you get also a nice introduction on how to run these tools in a cloud environment, for example, or in an HPC environment. And most importantly, these containers are able to be automatically consumed by workflow management systems like SnakeMake, Nextflow, and Galaxy, for example. And no matter which technology you use, if you want to use Singularity containers or Docker containers, or maybe pure Bioconda packages, all these technologies are actually made out of the same binary. So they are all reproducible across these technologies. The next step then was to annotate our tools. So the ELIXIR tools platform, and especially nowadays Hans from ELIXIR Denmark, is developing a registry where these tools are registered and where we collect a lot of metadata to these tools. So the citations and which input formats they can consume, which output formats, but also in which category they belong. So what was very interesting, but also very needed for us, was that we were going through all our tool catalogs and we're labeling tools with a COVID-19 tag. So we started this discussion on GitHub, and then we added support to bio.tools to tag tools with COVID-19 so that people can easily find it. So the findability of tools was here a key point, but also then that you can search for them or link out to a specific subdomain of bio.tools, so the COVID-19 subdomain, where you then in one page can see all the COVID-19 relevant tools. So the idea is here, if a user wants to search for a mapping tool that is very specific for virology data and so on, you can go to this website and then see, okay, these are the COVID-19 tools, which mapping or which variant calling tools are people using and are recommended for COVID-19 research. This is then how it looks like, for example. And as you also can see here, these are not only classical tools that you execute on a command line, but these can be also resources like the job Johns Hopkins Resource Center, which are then linked out. So you have one page with a lot of resources, intelligently grouped, which you can search and inspect. The next step, which we did, and this was also led by Nicola from the Great Britain ELIXIR node, is integrating tools into the Galaxy platform. So the Galaxy platform is an open source web-based platform for computational research with a special focus on accessibility and reproducibility. And there are already 7,000 tools available in one web page, so in one web server. And this goes from genomics to proteomics, but even to climate or material science. And what we have done, I mean, this was just one project out of many, but one of these biohackathon projects that we did, we're actually adding new tools relevant for COVID-19 research also to Galaxy that was not previously integrated. So essentially, we added a lot of tools for viral pangenome assembly. So in that case, it was VG and SegWish and so on. And these tools are now in Galaxy. And what we did in the end of the hackathon was then creating this workflow, which is still available in an updated version even now in different Galaxy instances, which you can use. And the tool which was integrated, or the tools that have been integrated, are now accessible via a web interface. So you don't even need to have a command line interface to execute your tools. You just go to this website and execute Minimap or any other tool for your COVID-19 research, and it will be executed on the cloud. If you have now, or if we have now these tools, or in that special case also the first workflows that we actually created, the task becomes, how do we actually expose those workflows to the broader community? So again, findability of workflows is a key ingredient that we wanted to work on. And in EOSC Live, we already had a project that we called Workflow Hub. This project is led by Carol Gobble and Frederick Coppens and the entire Workflow Hub Club across ELIXIR nodes. And the idea here is to have a workflow management agnostic system where you can register all kinds of workflows. And then they are searchable, but also groupable in groups. You can filter them. So pretty much what the bio.tool site is for tools, the Workflow Hub is for workflows. So here a lot of initial work was done, and the first workflows that we actually registered in Workflow Hub were actually COVID-19 workflows. So you can expect a first beta release this month. I wanted to stress the fact that this is really workflow agnostic. So we really tried hard to get Nextflow, SnakeMake, CWL workflows integrated in Workflow Hub as well. We will add more workflows as the time goes. But you are also encouraged to submit your own workflow or register your own workflows. You can see special COVID-19 workflows, so workflows that are tagged with COVID-19 related work under a special subdomain here. And this is how it looks like currently. So you have a depiction and a small image of the workflow. You have a description about your inputs, how many steps you have. We really would like to shoot out to the contributors, to the developers of the workflow, and provide a lot of metadata to the workflow, how you can render it. And the future idea is a little bit to get more metadata actually out of these workflow management systems, but also maybe link back to workflow execution engines. So for example, in the case of Galaxy, what we are aiming for is that if you find a Galaxy workflow on Workflow Hub, that you will have a button that says execute on whatever, the French Galaxy server or the Belgian Galaxy server. This is the roadmap of the Workflow Hub. So there's a lot that we would like to do. If you want to help us, if you like this idea, please contact us. There's still a lot to do, and we are eager to push out the first beta release this month. If you have now these workflows and these workflows are registered, or even before it gets essential to actually test these workflows and test also the execution of workflows. So I would just like to highlight a few projects that we have developed in the Galaxy community, which is also part of the tools platform. A lot of workflows to the genomic aspect, but also proteomic workflows that are processing and reanalyzing publicly available proteomic data set. But we had also a nice collaboration with the Diamond and Synchrotron people, where we developed a bunch of workflows for drug discovery and ligand-based drug design. And all these workflows are now in the Workflow Hub. But what we would like to do, or what we are doing here, is we really execute these workflows on a regular basis against public cloud infrastructures, ELIXIR clouds, or EOS based clouds, to actually ensure that these workflows are working and keep working over time. So you see here that we did that for Galaxy, and there's the NF Core community that does that as well. Just ensuring that if you use a workflows that we developed is actually working and keeps working. This is how it looks like. So you can define such a workflow text in a simple YAML file. And in the case of Galaxy, for example, you can use a command line tool to execute these workflows against a given Galaxy instance. And test that the expected output actually fits the output that you are now getting. This all can be hooked up to a CI infrastructure. So this.
Bjoern, we're running out of time.
Yep.
Sorry.
Okay. The last piece which is missing is more or less the deployments of workflows. So here, what we have done is we have actually worked on, in the beginning of the summer, on executing these workflows on different instances across Europe. Also the European Galaxy server. So you can now run your cheminformatic drug discovery workflows with millions of jobs against this infrastructure. And this is, for example, possible because we also distribute jobs across Europe. So we actually submit jobs from one Galaxy instance to clouds in Europe. This is because we want to actually give you the opportunity to calculate millions of jobs. What we also did is we developed training material. We enhanced the training infrastructure for that. So you can check out COVID-19 related trainings. There's also a webinar that ELIXIR organized with the tools platform. So if you want to learn more about that, what I just told you a little bit in a web style and a video style, please check that out. And there are a lot of events going on where you can exactly learn that. And with that, I would like to thank everyone, and especially a shout out to the Open eBench people, which is all part of the tools platform. But I could not cover that. And but just imagine all tools and workflows, what we are doing are also now benchmarked. Thanks.
Thanks very much, Bjoern. That was a very comprehensive overview of the tools platform. Unfortunately, we haven't got time for questions, but if you want to post something in the Q&A, Bjoern would be very pleased to answer it offline. Okay, we're going to move on to the next speaker. So I'd like to introduce Abdulrahman Azab Mohammed. He's from the University Centre for Information Technology from the University of Oslo, so from our Norwegian node. And he's going to talk about, we're going to change gear from COVID-19 now to federated data, and he's going to talk about the Nordic development on federation of the EGA. Abdulrahman, can you present your slides, please? That's perfect.
Yeah, thank you. Yes, my name is Abdulrahman Azab. I'm the project manager of the Nordic TRIGVA project, that is Nordic Collaboration on Sensitive Data. And one key work package in the TRIGVA project is the Nordic development on federating the EGA. There are many authors here who contributed intensively to the success of this work. And we are ending the project by the end of October, so hopefully we are going to deliver good results. So here are some acronyms which can be useful for those who are going to revise the slides afterwards. I would like to introduce first the TRIGVA project. It's a collaboration of Nordic ELIXIR nodes in Denmark, Sweden, Finland, and Norway to develop and provide compute services for human data across borders. The main target for overcoming the challenge is to prevent non-authorized use of data while utilizing the data for the benefit of the society and individuals. And this is the vision of TRIGVA. TRIGVA develops and facilitates access to secure infrastructure for sensitive data suitable for hosting large-scale cross-border biomedical research studies. In TRIGVA, we are technology developers, and we deal with mainly 4 infrastructures for analyzing and storing sensitive data. That is the ePOTA, which is the cloud infrastructure in Finland, and TSD, Tjeneste for sensitive data in Norway, and Computer-Rom at DTU in Denmark, and SNIC Bianca in Sweden. The cloud one is ePOTA, and secure HPC is TSD, Bianca, and Computer-Rom. Okay, so about EGA, European Genome Phenome Archive, very brief history, is a resource for permanent secure archiving and sharing of all types of potentially identifiable human genetic and phenotypic data. And it was launched in 2008 by the EBI, and in 2012, EBI and CRG joined efforts to collaborate on the central EGA portals. And of course, data fairness is taking into account for sensitive data. So the main purpose is there are data owners and who owns the sensitive data, and they would like to publish some information, metadata about their sensitive data on a website, so that collaborators or researchers who are interested in this type of data can go to this website and find information about these data sets. Maybe they find something interesting, then they would like to get in contact with the data owners and say, can I get access to this data? And this is a decision to be made by the Data Access Committee. So this is the main workflow. First, the data owner submits the data securely to the archive, which is the central EGA, and fill in the metadata about these experiments, about the data set, anything that is useful, information that are useful and can be shared. And this will be published on the website. And it can be found by anyone who wants to search this website. Now the data is stored in the archive, and a requester who found some interesting information here would like to request access for this data. Then there is a secure way for requesting access and making a decision by the Data Access Committee to either grant or deny this access. And then the requester can get access or not. the infrastructure on the European Genome Phenome Archive. Now there are researchers from different countries who are interested in sharing their sensitive data in the archive in a secure way. If everyone is going to submit data to this central place, will it be okay according to the legislation? We have the GDPR, which is even more soft than local legislations in some countries, as we all know. So the central storage might not always work. Then it comes the new idea of storing only the metadata in the central EGA and having local storage in each of the sites where the data is stored there. So the data doesn't leave the country. We don't have problem with the legislation, but the metadata will stay in the central website that everyone gets access to, which will be perfect. Benefits for the research community: unified brand for storage, discovery, and access to human genetic and phenotype data through the central EGA, and unified accession and versioning scheme for identifying and referencing data conforming to the FAIR principles. So the metadata will keep unified on the central EGA, and the data will stay secure in the local storage. Connection to TRIGVA. TRIGVA Sensitive Data Archiving Work Package. We are supporting the needs for archiving sensitive data in the Nordics, and it originates from the activities in the ELIXIR Accelerate Work Package 9 on human data. And our main aim is to extend the functionality of the European Genome Phenome Archive by developing what is known as local archive instances, or local EGAs. And those are the local storage instances which are connected to the central EGA. In addition to enabling establishment of standalone archiving instances. Maybe some nodes, they would not like to join the EGA Federation. They would like to have their own instances of sensitive data archiving services that are not connected to the EGA Federation. One example of that is that Denmark have not yet decided to join the Federation, so they are going with the standalone version. So those are two options that the TRIGVA project provides. This is a simplified, sort of simplified architecture overview. So this is the central EGA. This is the portal, this is the website, this is the data base for the metadata, and there is the EGA AI for access, and there is also plan for supporting ELIXIR AI. So this is how the users authenticate. And then the local EGA, this will be in Norway, in France, and whatever, where the local storage is. The users, when they log into the portal, they fill in the data for the sensitive data record information, and this will be stored in the central EGA. And then they will upload through the same website to the local EGA, and there will be ingestion, verification, and the data will be stored in the archive. And when another user who will log in through EGA AI or ELIXIR AI would like to access this data, I like this data set, can I get access? We'll submit the request through the same website, and then through the same interface, we'll get the data out from where it is. So for the user, whether it is a data owner or a data requester, they will deal only with that website. So all of these layers are abstracted from the user, and for them, things will be very user-friendly, while this complexity is hidden in the back. This is the architecture overview for the local EGA. There are different implementations. This is the way how it is implemented in Finland, for example, and it is a bit more complex because they use the infrastructure as a service cloud, and this is how it is in Sweden. So they use a specific network here for storing the sensitive data archive in S3-based archive. And this is in Norway, which is even more complex. What I wanted to show here is that the local EGA is not just a piece of service that you have to install it on a specific infrastructure with specific configuration. There can be different ways of installing local EGA. It is microservice based, and all microservices runs as containers. And this is an example of the Norwegian local EGA. This is the website, and you can log into the Norwegian local EGA with ELIXIR AI and get the start using the service from there. Where we are now, we have demonstrated the standalone sensitive data archive, and there is a link here to a tutorial on YouTube, and also mirroring data and metadata from central EGA. If we want to install a standalone local EGA, then we want to have the metadata also stored locally, not only in the central EGA, and there is a link for the YouTube there. And this is the documentation for the Nordic sensitive data archiving services on ReadDocs. And I would like to endorse our partners, Trygva partners, ELIXIR nodes in Finland, Norway, Sweden, and Denmark, and central EGA node and CRG and EVI. And this is the contacts for the Trygva project, myself, the project manager, and the scientific manager, Henrik Zassi, and we have sub-project managers from each of the Nordic nodes with the contacts here.
Thank you very much, Abdulrahman. That was a very comprehensive overview of Trygva. I have one question from Jonathan Tedds. He's asking, what are the key lessons you'd pass on to other groups based on your experience in working with different partners using different legal systems in the Trygva project?
In the Trygva project, we have a package called LC package. That is ethical, legal, and social implication. It was a bit of hard work to get to trust each other, so that we get to a common understanding on the legislation. It took some time. We had our security officers from Finland, from Norway, from Denmark, and from Sweden having meetings. We made, I think, 3 security workshops and some legal training on GDPR. So it's important to start working together as soon as possible, because it takes time and effort, and not to work separately, because things can collapse afterwards. So saving time and effort by starting collaboration as soon as possible would be a good idea.
Perfect. We're going to have to move on because we're running out of time, but if anybody's got any questions, then Abdulrahman will answer in the Q&A. Thank you very much. So we're moving on now to our final talk in the session. This is from Flora from VIB in Ghent, from our Belgium node, and a very important area for ELIXIR is fair data by design. Please take it away, Flora.
Can you see my screen?
Yes, but it's in presenter mode at the moment. Yeah. If you can. Perfect.
Okay. So today I'm going to present our software that we are developing at ELIXIR Belgium. And this software should help researchers to make data fair by design. Indeed, fair is the goal, not only for funders, government institution, but also for researchers that value transparent and reproducible research. As you may know, FAIR stands for findable, accessible, interoperable, and reusable. And to summarize briefly this concept, we can say that findable dataset it's findable when it's well described and metadata are provided, and also when there is an identifier associated. Accessible refers to the clear explanation of who, when, and how can access the data, and usually accessible data are in public repository. Interoperables means that the data should work and run on different software platforms and systems. So of course, using common vocabularies or standards, so like ontology and open format will support that. And finally, for a dataset to be reusable, you have to apply clear license and documentation to make the data understandable. Despite fair data being the goal or the path that everyone would like to follow, it's true that in reality, most of us, or most researchers, cares about the data only at the end of the project, or when the reviewers or the editors ask the data, or if a collaborator asks about the data. So at that point, make data fair can be too late for many aspects because information could be already lost, documentation could be not comprehensible, and so on. So to avoid this issue, to address this problem, at ELIXIR Belgium, we decided to develop a platform which is called FreedomHub.be. And it's actually a Belgian instance of the open software SEEK, developed by the Freedom initiatives. With this platform, we would like to make to enable researchers to make their data fair by design. By design, meaning in the meanwhile, the experiment or the project is still in progress, so not only at the end. The idea behind this platform is that FreedomHub.be would be an interface between the repository metadata schema or checklist with the ontology, of course, and the biologists on the front end, so the life science researchers. So we would like them to use the repository's metadata, so in the checklist and all the attributes that are needed at the end when you share your data already at the beginning of the project. More specifically, FreedomHub.be use ISA format as a generic framework for metadata. And ISA stands for investigation, study, and assay. Investigation is like the context of the project. The study is more a specific biological question and hypothesis, and the assay is in fact the technique that you do. So for example, a title for the investigation could be P53 in lung cancer. A title for the study could be the DNA methylation in P53 gene, and an assay could be DNA extraction. The source material and study is where the biological materials come from, so the description of the source and the treatment that you apply. And the protocol is the step-by-step experimental protocol in the assay. And the output can be samples, but can be DNA, can be extract, can be digital file. It depends on what is the assay. So we would like to use this framework to map the attributes that come from the repositories, starting with the EBI repositories, especially ARIA-Express and ENA. So we take those metadata schema, we take the attributes, and we map them at the study level or at the assay level. An example of attributes of the study could be, for example, a tissue or organism, species, age, dose. Another example of attributes for assay stat could be instead a microliter of something, or the volume, or the concentration, or the machine that you use. So this kind of information. And we provide these metadata, so these attributes, to the researchers on the front end of the platform, just like a drop-down menu of a metadata schema, where we show the attributes in two different steps. So at the study design level or the assay level, because we try to ask to the researchers the right metadata at the right time, not all together or all at once at the end. So the biologists then can choose the attributes and design their study and make some kind of a template, let's say, so this list of attributes. And then we want to implement the feature of exporting this template so that the researchers can fill in the information offline. And also the feature of submitting, so directly to the EBI repositories. So a way to broker specific metadata and data type to the right repository directly from the platform. So now I will show a few screenshots just to show you our progress in the development. So the first screen is showing the structure of ISA, and also the user can create or explore new project. When you create a project, for example, ERC maze, you have to give certain details like the title, the description. Within project, user can create one or more investigation. For example, leaves growth, in this case, is an example about plants. And in the details, the user can set the sharing permission. Within investigation, the user can create one or more study. And here there is one study about the GA gene in a mild drought condition, for example. And it's important to know that in study, there is a specific tab that is the design tab. And it's there that the user will see a drop-down menu with the metadata schema from different repository, for example, for plant in this example, to describe the source material at the study level. For each metadata schema, the attributes will be shown, and also if there is a short name and a description, so an ontology associated. When the users select the metadata schema, then all the attributes will be added at the bottom as a column name of a table. So for example, organism, developmental stage, etc. When the design is saved, then a source box will be presented in this flowchart canvas. And then here the user can start describing the assays that he or she will perform on the source, and also in which order. To create an assay, it's enough just to drag and drop the assay box on the canvas, and then an assay window will open. And also here the users can select a specific assay type, which is related to technique, and the attributes of the assay type. So for example, here there's the one from AliExpress for library construction. The attribute will be shown and will be indeed a library layout, library source, so all the required attribute for this assay type in the repository. And the user can provide a title and should provide protocol, so with the step-by-step experimental procedure. Once the assay is saved, then the assay will be shown in the canvas. The assay will be also shown on the side bar on the left, and the attributes from the source, so the study level, and also the attributes of this specific assay will be shown in this table as a column name. And here the user can add rows and enter information for every observation unit of the study or for every sample. So every row will have all the attributes specified for source and for each assay. So here, for example, for library preparation, we'll have library layout. So to summarize, with this platform, we aim to input the metadata schemas and attributes with the ontology from EBI repositories in the backend of our software, so that we can provide the right metadata at the right moment according to the ways of working of the researchers to the users, and the user can fill in the information, so the data and metadata, and then submit it through the platform to the public repository like EBI. With this, I have to thank Frederic Koppels, if you don't know, of ELIXIR Belgium, Farid Kiani, which is our software developer, all FAIRDOM Association, especially Carol Grob's group, and Stuart Owen, and our funders, and of course, for your attention. And I'll be happy to take any question or suggestion. And I just say that we are in at the Biohackathon as well. So if you would like to contribute and give suggestion, we are very welcome.
Thank you, Flora. That was a very comprehensive overview of FAIRDOM. I don't see any questions at the moment, so I'll have one. What is the major advantage for the biologists to actually engage with FAIRDOM for you? What is the major incentive, do you think?
So as a short-term incentives, we would like to give indeed the opportunity to make it way easier sharing the data in the public repositories like ENA on AliExpress. So especially now people are really seeing how important this is. Then if you already have, you organize your data in the platform to begin with, then at the end, it will be very easy to share your data. Plus this huge advantage that everything will be annotated properly according to ontology. So there will be no doubts that you will remember what you did 2 years before. And this is also important. And so we don't have any data loss or metadata loss in the lab.
Okay, thank you. We have to wrap up this session now as we're running out of time, but I'd like to thank all the speakers for very informative presentations and for keeping on time as well, because that wasn't an easy job. So thank you very much. If anybody's got any questions, please contact the speakers. They'll be happy to feed back. And now I'll leave it for the ECCB organisers to take over for the last plenary session. Thank you very much to everyone.
Thank you, Jen.
Thank you all.
Thank you all.
Everyone, and see you in 8 minutes in the next session. Thank you. Bye-bye.